Uh, just a quick question. Let me know if you can hear the video. Can you hear it?
No.
Oh, you can't. Oh, how can I?
Can you type the URL into into the chat? Because I can do it off of my screen if nothing else.
Okay, yeah, maybe I just let everyone uh watch the video for 3 minutes. Okay. Stop sharing. I think it's an amazing video about the history of the city and the fall.
Why are photographers ditching their expensive cameras for this
All right. Zoom meeting, share screen. I have all these little pop-ups that are coming up.
Oh yeah, I have that too.
All right.
NASA super zoom lens? Because it clips on.
Ten 10,000 years ago, you might have seen this glacial river Warren Falls in St. Paul, this massive waterfall that would have looked something like Niagara Falls here. And with 1 million cubic feet of water pounding over it down into a valley that was 100 feet deeper than it is today. And you might have seen woolly mammoths, walking along the edges of that canyon and that waterfall with American Indians, trying to maybe hunt them down.
The incredible volume of water cascading down Glacial River Warren got its start from Glacial Lake Agassiz. This huge lake was formed by melting glaciers at the end of the last ice age. The glacial river carved a path that you'd recognize today as following the Minnesota River and then the Mississippi downstream from their confluence. It has been a particularly restless waterfall that has migrated upstream because of the geological process you can see at work today. Glacial River Warren flowed over a surface layer of limestone on top of softer sandstone. Powerful recirculating currents at the base of the falls eroded the sandstone faster than the limestone above it. Without the support of the sandstone, great chunks of limestone collapsed into the river below the falls. In geological terms, the falls raced upstream at a breakneck pace. About 11,700 years ago, it was near the site of downtown St. Paul. 2,000 years later, the falls had moved upstream past the confluence of the Mississippi and Minnesota rivers and arrived at the point where Minnehaha Creek joins the Mississippi. The main waterfall continued up the Mississippi as the same process of erosion carved the deep gorge visible today below Minnehaha Falls. A close look behind the veil of water at Minnehaha Falls shows the two layers of bedrock, the harder limestone on top and softer sandstone below, which continue to erode each at its own pace. The main waterfall had arrived at the site of what was to become the city of Minneapolis. When it was given the name St. Anthony Falls by Father Louis Hennepin in 1680. The falls eventually gave birth to Minneapolis, providing power to the sawmills and grain mills that were the city's first industries. Today, the apron of concrete prevents further erosion of the falls, ending their 15-mile upstream journey. A journey that was undertaken at the breathless pace of 6.8 feet per year.
Awesome.
I did not know that.
It is a man-made fall, and I think just the fact that we made it here, it's how the Twin Cities get developed. Have later we have so many meal factories and And St. Anthony Falls Laboratory is actually one of the best in like geophysical processes, like how sediment moves, how channel migrate. We used to be a National Science Foundation research center. Like I did my PhD at MIT. I studied in Princeton. I exchanged in Caltech. But I think in terms of facility and geophysical process, Stanford is the best. Like among all the universities I know, and oftentimes we kind of ignore that. Like the geomorphology processes in our daily life, because we live in a city in concrete, but our river are always migrating, and they cause a lot of issues. Like when river migrate, they can cause erosion, they cut the banks. So there are a lot of consulting companies to look at how this river migration can cause issues. For example, there was one river that is migrating that may cut the highway, cause a lot of trouble. And there was sometimes when people try to build a museum in a mountain, they realize the river may migrate, so the whole area can be eroded. So there are like thousands of people studying sediment transport erosion for like centuries, like over a century. Yeah.
Wow.
Okay. And
all right, we're gonna turn it over to you and let you start talking about the nitty-gritty of your research.
okay, yeah, I'll try to keep it brief. Thanks. Sorry. Yeah. Okay, can everyone see the slides?
Yes.
Okay. So as all of us has experienced through COVID, I learned an important lesson from COVID, which is when tiny stuff, especially biological stuff, starts to move, the world can be chaotic. So my research is about how tiny things such as microbes and clay and other biological stuff such as vegetation, how they interact with our aquatic environments like rivers, coastal systems, and how they shape our Earth surfaces. And there are 4 research directions in my group. The first is erosion in rivers and coastal areas. So erosion is such an important issue that has been studied for over a century, but the majority of equations are developed for non-cohesive sediments such as sand, because it's just relatively easy to study. But people already realize that the erosion of the cohesive mud is completely different from the sand, because you have this clay mineral, they have surface charge and they are nanoparticles, they aggregate. They completely change the aggregation, erosion, the property of the soil. And so does these microbial biofilms. They produce this organic matter which can glue things together, change their property. So whenever we have clay and a biofilm, the erosion can be changed by several orders of magnitude, but we don't have a clear idea of how to predict it. So this is something my group is devoting our significant amount of time on. And the second research question my group is interested in is harmful algal blooms. As many of you already see in a lot of Minnesota's lakes, harmful algal bloom is a significant issue in the Great Lakes region. Each year, there are over several hundreds of millions of dollars lost due to harmful algal blooms, and a lot of animals, dogs get sick, and even people. And one of the way to remove harmful algal blooms that has been used in East Asian country is to use natural clay to spray it on the sediment, and which cause harmful algae cells to flocculate, aggregate and sediment to the bottom. While this method has been used in East Asia and now in Florida, it hasn't been adopted in Great Lakes. So we have a project to look at how we can use clay to remove harmful algal blooms. And also my group is interested in how chemicals and bacteria move in soil and how they generate carbon, which can change our climate. For example, this is red, the yellow colors indicate where the bacteria are and how they use carbon. And we also look at how carbon is sequestrated in wetlands. Finally, my group is also looking at how bacteria grow in addition to soil, such as in teeth and tissues. So you may think that these are completely different research questions. However, they all share a lot of commonality. First, the interactions between the particles, microbes, and the fluid play a critical role in these issues. And the interaction spans multiple scales. So what happens at this tiny micro scale that was very hard to see in our eye actually controls the performance of the whole system. So we want to know how can we understand these issues across multiple scales and across physics and biology. The uniqueness of my group is we are one of the few, if any, lab that have technology across different scales and involve physics and microbiology. So at the larger scale, in terms of meters, we do flume scale experiments to look at sediment transport and the exchange between surface water and groundwater. For example, here we use this vegetation dials to simulate vegetation. We put sediment into the channel and visualize how sediment moves. I'll talk a little bit more later. And at the middle scale, we do our own imaging system to look at how, for example, this green fluorescent bacteria, how they spread in soil. And we make our own imaging system to look at how these harmful algal cells at the bottom, these green symbols, can you see my mouse? How they flocculate, how we can use different materials to cause them to sediment. And we also have microscale microfluidic technology, which are these tiny channels. Their diameter can be less than the diameter of our hair. So we fabricate these channels in the clean room, which is like a super clean room. There's no dust in the air, and we have to dress up in suits to cover ourselves and make these chips. And then we can inject bacteria, inject particles to look at how they interact. For example, here you can see the circles are just glass beads, and these green symbols are the bacteria. So we were able to visualize bacteria that are micron scale, way much less than the size of diameter, size of our hair in these microfluidic chips and with a very fancy microscope. So now I'm going to briefly talk about a few projects in my group that involves different technology, different scales. So the first is about the impact of clay and the biofilms on bacteria, on erosion and adsorption of organic matter. So the first question is, can we be able to understand how this tiny bacteria develop these biofilms on surfaces? So biofilms are just aggregates of bacteria. So bacteria, in a lot of cases, they're not single cells. They are cells aggregated together, and they produce organic matter that bind them together. So we want to know how bacteria exist on surfaces and how they cause sediment to aggregates, which change their transport. So in this case, we actually fabricated this microfluidic chip. This is 50 microns. Consider our hair are hundreds of microns. So this can be less than half the diameter of our hair. And we can design different patterns, and then we can visualize the development of bacteria, biofilms. So these gray colors here are biofilms on these surfaces. So here is an example of how this biofilm, this gray color, are developed on the flat surface. And then we were able to trace the boundary of the biofilm and measure the biofilm thickness and study how this biofilm were formed over time under different flow conditions. So this different color indicate the flow rate or the shear stress in this channel. As we can see, if we increase the shear, they actually prevent biofilm growth, which makes sense that if we flash something very hard, the bacterial biofilm can break out and stop growing. And we also did a lot of simulation. So the colored image are the flow field, the velocity field inside the channel. And we compared with our measured biofilm, and we found out the fluid mechanics, the flow actually controls the biofilm development. And the one thing very interesting is that we found the boundary of the biofilm correspond to the flow velocity, which is similar to bacteria swimming speed, which suggests how bacteria moves actually controls how they colonize surfaces and how they form biofilms, which is something that hasn't been understood yet. So people always think biofilm has little to do with bacteria swimming. So to understand this, I'll skip this theory part. So we do a lot of fluid mechanics theory and compared with experimental data. I'll skip this part. So to understand how swimming controls biofilm growth, we actually did high-speed imaging. So we can take photos like up to 200 frames per second. And then we were able to trace the movement of individual bacteria. So here shows how a swimming bacteria move towards the wall and form biofilms. So this bacteria are just like a couple microns. And then we did experiments where we genetically modified the bacteria, so they were no longer able to swim. And so we can see they just being advected away by the flow. They were not able to attach to the surface and form biofilm.
Dr. Yang,
So those that, yes.
are you
Okay.
tagging the bacteria with something like a fluorescent probe?
So here we didn't use fluorescent probe. It's
Okay.
just because the reflective index of bacteria and water is different. So even
Sure.
just use non-fluorescent camera, we can able to see it. Great question,
Okay.
thanks. Yeah, we can use fluorescent probe. Yeah, but for this case, we just use a regular bacteria. Yeah. So we do a lot of things on biofilm, including how if we have fluctuating flows, because in nature the flow is not constant, it always fluctuates back and forth, and how that can affect biofilm growth. So in this study, we compare the biofilm growth under steady conditions. The biofilm have 4 different phases. So then when you start to grow biofilm, they may sometimes adjust lag phase, and then they grow exponentially, and then they reach a plateau, and then they decline. So this is when we have a constant flow. But then we add fluctuating flows, and you can see the biofilm. So this is the thickness of biofilm. You can see the biofilm start to fluctuate with the flow. And the mean biofilm thickness actually start to increase. And then we increase the frequency, make the flow fluctuate more and more frequent. And then you can see if we have high fluctuation, they actually suppress biofilm growth. So this is example showing like low frequency biofilm growth get promoted. But if we have high frequency, the biofilm growth get suppressed. So what we found out is that the bacteria, when they adjust the flow, when we change the flow, they need some adjust time to grow. And this adjust time is what caused this phenomenon. So the fact is, when we have high fluctuation flow, because biofilm, they need some adjust time, T0 to grow. When you fluctuate too fast, the biofilm doesn't get enough time to grow, which is why high fluctuation flow can suppress biofilm growth. So we were able to develop some mathematical model that can able to predict the growth of biofilms under different flow fluctuation frequencies. So here, the solid line are the biofilms we predict, and the dashed line are our experimental results. So those studies of how biofilm grows on surfaces provide us a framework to understand how biofilm grow on surfaces, which can glue sediment together, change transport. But this study also provide a lot of information of how biofilm grow, for example, on our teeth, which cause dental plaques, and how they grow on our lungs, our tissues. So all this are related, and those fundamental work can help us understand and address a wide range of topics. So right now, my students is working on how biofilm actually grow around particles under different flow conditions. And you can see these beautiful streamlines get formed under different flow conditions. And we were able to show that the physics do play an important role in shaping the microbial community and the microbial shapes. For example, if we increase the flow rate, you can see the biofilm streamers form different morphology. And right now, we are looking at how this biofilm glue these different particles together, which can change their transport and migration. Okay. So those are the formation biofilm. After biofilm formed, and those bacteria can also start to interact with the chemistry in the soil and in water. So we also have projects looking at how bacteria use the organic matter in soil, which can control CO2 emission from soil. So soil actually contains way more carbon than all the carbon in the atmosphere. So there are a lot of concerns of whether future climate can cause the carbon organic matter in the soil to decompose it. to like decompose and then emit carbon. However, soil is translucent. It's very hard to say what's going on inside the soil, right? Like what we can do is measure the amount of CO2 emitted from soil under different temperature and guess what's going on inside the soil. So what my group has pioneered in the past is we decided that we are going to do experiments, do synthetic experiments in a soil on a chip using microfluidic technology. So we found a transparent clay that is similar to natural bentonite clay with similar molecular structure. But because it doesn't absorb the visible light energy, so it appears to be transparent. We put it in a microfluidic channel. We put fluorescent labeled bacteria and fluorescently labeled enzymes and organic matter and look at the absorption of organic matter into the clay and the desorption processes. Okay, so we try different amount of sugar. So this is the example showing how the green sugar gets absorbed in a clay that is about 60 microns in size and how it gets washed out. And as we can see, if we start to wash the clay, a lot of organic matter gets sequestrated in sediment. So we were able to see how organic matter get absorbed and sequestrated in soil using our technology. Okay, I'm going to skip these details. And then we also looked at how bacteria interact with the clay. So here the green color is the clay absorbed with green organic matter, and the red dots are the bacteria. So we're able to see that how the clay actually protect the carbon from bacteria decomposition. And we did a lot of study. For example, this is simulate a soil pore, and we inject different sugars and look at how sugar get absorbed into the transparent soil. And then we inject water, nothing happened. We inject enzyme, we can see how the sugar get chopped down and released by enzymes. Okay, I'm going to skip these details. So in addition to this micro scale, microfluidic experiment, we also do mesoscale experiments. For example, we look at how bacteria spread in soil. And one thing unique about the soil is a very complicated. You can see we have grains. When two sediment grains attach, two sand touch, we form these corners. We have air and we have water. So we have air-water interfaces. Sorry, I'm getting water. However, how bacteria, how they move in soil isn't fundamentally understood. Soil is opaque, it's very hard to say what's going on inside. So I did something very simple. I fabricated these chambers. This is just transparent chamber. You can see a prison-shaped chamber here. And we made different angles. And then we put this bacteria with green color in this chamber and start to grow bacteria in it. And this is one chamber with different corner, 60 degrees, 30 degree, 90 degree. And here is the video capturing the bacteria, the green bacteria in this chamber. The top black color are just air. The green color just bacterial solution. And over the course of a day, I've discovered something very interesting and crazy. It's just, okay, the bacteria, they actually self-generate flows along the sharp corner, but not at the 90-degree corner, and a weak flow at a 60-degree corner. Can anyone guess how? So in control experiments without bacteria, nothing happened, only evaporation. So can anyone guess why do we have these flows at the corner? Like when you put bacteria in a chamber, they just generate this flow along sharp corner for like hours.
Does it provide them with maybe some stability?
You mean the corner provides some stability?
Yeah.
That's a great idea, but maybe not for this case. Like when there is a movement, right, there is a force. What is the force? What is the reason that there is a force that generates this movement? Yes.
Is there
So-
an opening near that top part that's allowing air suction?
So everything is open to the air. So even the material we use is gas permeable. So everything is saturated with air. Okay, and I just want to, and these are bacteria. So they are soil bacteria. They are able to swim. They are able to form biofilm. They are able to attach to surfaces.
Adhesion?
So the idea they're swimming to the edges so that they can attach to each other.
Great idea. Close. So I was have the same thoughts as well. I thought, oh, maybe bacteria swimming or like adhesion attached to surfaces can generate this flow, right? So I test that idea. So we actually trace the movement of the tip using this bacteria that have pili attached to surface and have flagella that can rotate, which help them swim. And then we can see the flow was generated around 6 hours and then lasted for many hours. And then we use the mutant that doesn't have flagella, so they no longer swim. The same flow was generated. And then we use the mutant that doesn't attach to surfaces. The same flow was generated. So what's going on? We actually tried different mutant. We found out that the key to this problem is actually a chemical called biosurfactants. So surfactants are the kind of chemicals that we use every day. For example, our soap, our hand sanitizer, they can change the surface properties of air-water interface. And we found out that the key is because when bacteria generate these surfactants, they can change the contact angle of the liquid on the surface, which can generate a force along the sharp corners that generate this flow. So we were able to develop theories that based on this contact angle and the corner angle to predict at what situation bacteria can flow. And this great with what we observed. So our theory predict 60 degree, the critical angle, and we observe flow at 30 degree, but not at 90 degree, and a weak flow at 60 degree. So this is a new mechanism we discovered, and this shows how powerful our biology world and physical world are connected. So oftentimes people treat these two systems separately, but what we realize is our biological system plays such an important role in our physical system. When we have bacterial biofilm, they can change the erosion, the transport, the morphology of our rivers, channels. But at the same time, our physical system is changing our bacterial behavior. When we have flow, bacteria can grow at a different rate and form different biofilm. And bacteria can generate chemicals that change the physical system. For example, in this case, they generate biosurfactants. And then this chemical can generate flows. So I think to understand our natural environment, we do need an integrated and a coupled view to understand this system. So to further understand how this mechanism of bacteria generated chemicals can affect bacteria spreading in natural soil, we actually made this synthetic translucent soil, and we put a bacteria solution on the bottom, and we image it how bacteria spread into the soil. We use two types of bacteria. One is that can produce surfactants. One is that the one that doesn't produce surfactants. And as we can see, when bacteria can produce surfactants, they can penetrate into the soil. But if not, they will not be able to penetrate into the soil. Okay, and another example we have. Okay, I'm going to skip this one. Okay, so again, this is showing how the interaction between microbes, chemistry, and physics are very important to help us understand how bacteria spread in soil. And this mechanism also have important issues or like how disease, how pathogens are spreading in lungs, tissues, and our teeth. And another project I want to briefly talk about that my group has doing is how this micro scale and meso scale processes actually regulate larger scale processes. There are three projects, one about cohesive sediment transport, one about hyporheic exchange, one about harmful algal blooms, I'm going to briefly cover. So our next project is about how tiny clays, clay particles erode. One thing about clay, they have surface charge. So the chemical interaction actually plays a very important role in the erosion of sediment. So we measure the critical shear stress, which is the shear stress above which the sediment starts to erode under different salinity for this clay. So here is actually a 1 meter by 1 meter by 1 meter oscillating grid. So this grid is moving up and down to generate a shear stress, a force at the bottom. And then we use the imaging to observe when the sediment starts to move and then identify the critical shear stress. So we gradually increase the shear and identify the critical shear stress. On the right is the water recirculating channel, and we generally increase the water recirculation rate and then observe when the clay starts to erode. So this is the critical shear stress we measured on a different amount of salt in water. As we can see, if we add different amount of salt in water, the critical shear stress to erode the clay can change by almost 2 orders of magnitude, which hasn't been represented in current sediment transport models because most people only consider sand. And what we further find out is that this behavior is related to the microscale structure of clay. So at low salinity, clay can actually feel like a jello-like structure. So if you look at a microscope, it looks quite uniform. So these dark circles are just bubbles. So this clay, when they form a gel, they can hold hand with each other. But when they start to form this, at high salinity, they start to aggregate from this microscale aggregates instead of a gel. And this different behavior at the microscale actually determines its critical shear stress. So this is one example showing how microscale structures and behavior actually controls larger scale behavior. And currently, we have project to look at how bacterial biofilms affect the erosion and the formation of bed forms in channels. So here at Suffolk, we have a lot of channel to simulate rivers. Here is a 20-meter long channel. We put sand in it. We generate different flow conditions. And then we have laser scanner, acoustic measurements that can scan the whole topography, look at how sediment get moved, eroded. And then we spray this cohesion, this bacterial biofilm material on the sediment. And we found out that once we have this bacterial mat, we can generate different structures. So without biofilm mat, you can see just ripples, right? Once we have mat, it forms distinct structures, actually very interesting structures that are observed in rock records. So this is an example showing how understanding of these processes can actually help us interpret our geological records and the beginning of our Earth. Because when bacteria start to form biofilm, we can see our Earth starts to become different. Our soil become different, more rich, and then plants can start to grow. And I also previously have some summer student looking at how bacterial biofilm can affect channel morphology. So these are some preliminary results. So she actually ran experiments with a straight channel first, and then just let the channel evolve. After several hours, you can see the channel become braided. And at another experiment, she add the cohesive material, simulated bacterial biofilm on the channel. And you can see the channel doesn't form this morphology anymore when we have bacterial cohesion. A direct evidence of how this tiny cohesive material and a tiny bacterial biofilm can impact larger scale morphology. Feel free to stop me if you have any questions. And another question we can do in our lab is to look at how we can use different material to cause cells to aggregate to remove harmful algal blooms. So this is a project which we put different amount of material, leponite, into the water column and see how they can cause the harmful algal cells to flocculate and remove. So we actually recently had a paper which we discovered a new material that can successfully remove harmful algal cells, and we are going to test it in many of Minnesota's harmful algal bloom contaminated lakes this summer. So if you're interested in this project, please let me know. Yeah, so these are different materials, how their efficiency in remove harmful algal blooms. This paper has been accepted already. So we are combining this imaging, microscale imaging, and this larger scale flume experiment. So we have flumes that are tens of meters long, and we can grow harmful algae in them and then we can spray clay particles and use different methods to try to remove them in the lab so that we can able to apply it in the field in the future. Okay, I went through a lot of projects very quickly. I'm going to lastly talk about also very important topic in my group is about the exchange between surface water and groundwater. As we know, this exchange is called hyporheic exchange. It actually plays a very important role in our aquatic environment because we have a lot of contaminations in water. So if surface water get into groundwater, they can contaminate our groundwater and vice versa. And there are a lot of structures such as vegetation, log jams, fallen trees. They can impact this hyporheic exchange, but this effect hasn't been understood yet. So what we do in the lab is we actually made a transparent wetlands. So these are hydrogel beads we made in the lab are transparent. And then we use acrylic dowels to simulate transparent wetlands. And we inject this fluorescent dye, which emit green light when we shine blue light on them. So we were able to trace where the dye are, how they move, how far they move, and be able to quantify hyporheic exchange. So we have shown that vegetation actually plays a very important role in hyporheic exchange and develop some models to predict it. And we also build this transparent system with log jams. So this in the middle, these are just simulated piles of trees, which we call log jam. And we did a lot of velocity measurements using particle image velocimetry. We were able to trace the movement of the water inside the particle. This is a some example. So this is a log jam. The water is from left to right, and then we inject a dye into the sediment, transparent sediment. So we were able to trace the movement of the water into the sediment and how they move out. And then we were able to develop some predictive model to predict how, if we have things blocking in the channel, how that can increase the exchange between surface water and groundwater. Okay, so I just briefly went over a lot of projects in my group to show you that we have technology across different scales. We have a lot of flume experiments. We have to look at erosion, hyporheic exchange. We have mesoscale imaging system to look at how things spread in soil, harmful algal blooms. And we have microfluidic experiments to look at how biofilms grow, how bacteria moves, and how carbon gets sequestrated in clay. So our final goal, which may take a lot of years, is to combine these two technology together to understand different ecosystems and to address many important society issues such as coastal erosion, harmful algal bloom issues, climate change, and bacterial contamination. And we also do a lot of field work. So if you're interested in being part of this project and learn some of the technology, feel free to visit my lab and talk with my lab members. And with this, I would like to thank you for my group. So those are photos taken at the San Anthony Falls lab. And yeah. Thank you so much for your time. Let me know if you have any questions.
The amount of physics and engineering is impressive, like, and just all of the little tools and stuff like that. And the fact that you have like real world applications almost immediately, looking at those algal blooms in Minnesota lakes. And I think that's fascinating. So thank you.
Thank you.
Do you have anything in mind for the students and what they would be working on, or?
Yeah, so I have a postdoc just joined my group from Princeton University, and he is Avery. He's leading a project to use clay to treat harmful algal blooms. So we're going to do a lot of field work, go into a many pond, collect a lot of bucket of harmful algal bloom contaminated water, and test different clays. And I think that this project can have real impact on the state, because currently, to my knowledge, they're just not being treated. If the harmful algal bloom is not treated, they're just going to doubling, producing more toxins. So we're collaborating with the city of Minneapolis to test different methods, and hopefully we can get something running in the future. And this is one project. Some other projects are related to hyperic exchange, like doing some experiments on hyperic exchange, and some experiments related to channel morphology, like how channel migrate, like what you said in the video, doing some flume scale experiments. Micro scale experiments is very difficult to do in a short time because it takes a lot of training to go to the clean room. But these flume experiments can be easily can be learned in a relatively short amount of time.
That's great. Students, do you have any questions, thoughts?
I think the one question that I had is, once the algal blooms are brought down into the sediment, right, in the bottom of the waterway, what does their growth look like? And like, I know agricultural runoff is sort of what feeds these organisms, especially like excess nutrients from fertilizers and things that end up in the waterways. So if they're in the bottom of the waterway and they're getting plenty of sustenance, can they grow enough to come back out and get back into the waterway?
That's a great question, and something need to be verified. But my understanding, we did some preliminary results, is that once we use clay to flocculate the algae cells, the algae cells are actually encapsulated in clay, so they won't be able to grow. That's what we believe based on preliminary results, and I think that's something we definitely need to test in the future.
So we can look at the clay like carbon capture, right? Once it's in, or look at the algae.
Yes, exactly. That's a great question, because my postdoc, Avery, he's very interested in carbon capture as well. He had this idea of using clay to capture algae as a carbon capture.
Yeah, that's really interesting. And I, for lack of a better term, I'm just dumbfounded by your research.
Thank you. Thanks.
Other questions for Dr. Yang? No? All right. Well, I want to thank you for your time, Dr. Yang. Your work is fascinating. Liz, did you have any questions?
No, I just would like to reiterate that I really enjoyed your talk and your work is really awesome.
Not only my work, it's the team. The team is great.
Yes.
I'll
Your
let them
lab.
know your comments. And yeah, I believe most people here will visit Suffolk in July, and really looking forward to see you all in person, because right now I just see the names.
Yeah, we're excited. So July 3rd will be our site visit to St. Anthony Falls. So that'll be an opportunity for the entire group to go and see the facilities there. And yeah, I'm excited.
Yeah, me too. Same here. Yeah, thank you. Good to see
Right.
you. Yeah.
Thank
And
you.
yeah, if you are any interest in any of the project, feel free to get in touch with me, and have a great day.
Okay.
You
Take
too.
care. Thank you again.
Bye. Thank you.
Bye. All right. Well, we have a few minutes before Dr. McGovern joins us. We've heard 3 research talks so far, all of them in fairly different areas. So, think about what areas you're interested in, think about what you mesh with, think about, because after Dr. McGovern's talk, Liz is gonna hand out that survey for you guys to kind of rank your choices. For engineering people, or environmental science people, this project looks amazing. I did not understand all of the physics associated with that, so hopefully I'm not alone, and we have the graphs with the different symbols and yeah. But maybe that's your area and maybe you were like, oh yeah, that makes total sense, or oh, okay, that's interesting. I was just like, I have no idea what that means. But yeah, other thoughts about choosing labs, Liz? Or
No,
any
we'll
com-
talk about it. I'm also working on resources for next week that will include all the location information that you're going to need to get to where you're going next week. So yeah, I'll share all those details after we hear from Dr. McGovern today.
Okay. All right. On Monday, you guys are gonna come and meet me at Century College at 9 a.m. We're gonna do some lab safety, PCR, some bacterial cultures. You guys can do gram stains if you want. I've got that up and running, and I've got a gram negative and a gram positive culture ready for you guys. I had my kid help me with some environmental samples for bacterial cultures for you guys to like look at and practice on. He really wanted the inside of his baseball glove done. I'm terrified of what that's gonna look like, but you know, it'll be fine. It'll be fine.
That's really interesting. That'll be fun.
Yeah, just to kind of get some different, because there are some different types of bacteria that grow in different ways, and I can show you pre-cultured bacteria, and it looks very nice, and it looks kind of the same when you look at E. coli versus staph, but some of those environmental samples get really fun. So yeah.
So on Monday, you're sort of gonna have like a lot of different options of things that we can pick to
I'll kind
do.
of run you through them. Like we're gonna go through and you'll do each of those. I just have some extra stain. I mean, we're in a micro lab, so I've got all of that stuff together. And sometimes it's really nice to visualize and at least do some staining.
Yeah.
But you guys are gonna make gels that you're gonna run the next week. You're gonna do your PCR reaction. We'll do lab safety. And you guys are gonna start with like, I'm just gonna put all the PCR stuff out and give you the recipe and I'll walk you through it. But it's more like a graduate school lab where you have to do each step rather than as the instructor, usually in a bio lab, you would get everything handed to you, you know, and like this is prepared solution and this is this. And I'm gonna basically just set the thing of agar out and you guys have to weigh it out and make agar solutions and
So you've already inoculated the bacteria
yeah, but I
from,
can and you guys can transfer bacteria and see, you know, from one plate to another, and we can look at it next week and how that looks. And I can also send plates home if there's anything you want to look at. I will warn you that water fountains, you do not want to look at what that looks like at the end. Because you might stop using the water fountains. So there is some deniability that can happen with that. Yeah.
Do you need any help on Monday?
I don't, but I mean, welcome, Dr. McGovern.
Hey, everybody. Thank you for having me. My voice is a little hoarse. Sorry. Not sick, but I did fly to Boston. I'm from Rhode Island, speaking of where people are from. I'm from Rhode Island originally, and you know, the Celtics are in the NBA Finals, and I couldn't pass up the opportunity to go back and cheer them on. So I cheered a little too vigorously, and so my voice is still suffering for it. So sorry for that, but hopefully it's not too bad.
We can hear you just fine.
Okay, great. So my sense is that you guys wanted me to just kind of talk about opportunities in the lab, right? Is that right, Jessica?
Yeah, and if there's any like background information that of work that you did related to that, please feel free to share.
Yeah, okay. So that's what I'll do. I have kind of three main, I guess, branches, I would say, of work in my lab that are kind of related, kind of not related. It's stuff that I'm interested in and stuff that I've gotten interested in over time, and just this is kind of where the work has gone. And so what I'll try to do is just give you background on each of those branches, basically, and potential ways that that you could get involved if you want. You all can kind of feel free to stop me at any point, and ask me questions or whatever as we go. And let me just get my little laser pointer here so that way I know I can. You guys can see that little guy, right? So those three branches are basically. So I'm just by background, I'm a neurosurgeon at the university here. I'm what they call a stereotactic and functional neurosurgeon, which means, who knows what it means, but it means that it's a branch of neurosurgery that is kind of focused on neuromodulation, putting electrodes in the brain for a variety of different disease processes, including Parkinson's disease, tremor, epilepsy. Epilepsy is really a whole field actually unto itself, which involves putting electrodes in the brain or actually resecting brain that we think is causing seizures. So that's where a lot of my work is focused. And so the three main kind of branches of the work in my lab are focused on optimizing the deep brain stimulation electrodes. So not necessarily where we put them, but how they work. So I'll talk about a couple of projects related to how deep brain stimulation works in epilepsy and in Parkinson's disease, and some of the work that we're trying to do to make that better. I'm also interested in health equity and the fact that a lot of our neuromodulation procedures are really kind of inequitably distributed, right? If you look at a lot of the national data, and so doing some work within our own system to try to change that, basically. And then the third thing I'm very interested in is gait, so like walking and balance. And that is because the disease processes that we treat, especially in Parkinson's disease and this other disease called normal pressure hydrocephalus, it's really, those two things are really can be devastating to patients, and they're very poorly treated. So our current ways of treating, either with medication or surgery or anything, don't really work very well. So I'm very interested in trying to figure out how Parkinson's disease, how hydrocephalus affects people's walking and their balance, and how we can make that better. So I'll give you kind of just a brief walkthrough of each of those and how people can get involved if they're interested. So just starting with epilepsy. So epilepsy is pretty common and affects about 3 million people in the United States, about 3.5 million people, about 1% of the population. 2/3 of those people who have epilepsy, and epilepsy is really defined as having continual, recurrent, unprovoked seizures, right? 2/3 of those people can be controlled by just taking medication, but about a third of all of those people will continue to have seizures despite medication. So that's about 1 million people, right, in the United States. All of those people are potential surgical candidates, basically, and should go through a surgical eval. deep brain stimulation is basically a form of neuromodulation for people with epilepsy. It's not the first line treatment, because the first line treatment for epilepsy, in terms of surgery, is trying to find a certain spot in the brain where the seizures are coming from, and take that out, basically, because that's by far going to be the most likely to make somebody seizure free. And that's the goal in folks with epilepsy, is to become seizure free. So neuromodulation is not a first line treatment, but it is a pretty frequent treatment, because it's hard to find a very focal spot in the brain that is both causing seizures and can be taken out in everybody. And so, and the folks that you can't do that, deep brain stimulation or putting electrodes into a certain part of the brain to try to modify the networks of the brain is a treatment. And unfortunately, very few people will become seizure free from that. And we're really looking at improvements in seizure frequency, improvements in the severity of the seizures. Very few people will become seizure free. And so there's a treatment gap there where we can and we should try to do better. Part of the issue is that there's a randomized controlled trial that looked at this and found that it's more effective than just a placebo. And they use a certain set of settings to treat these folks. So they used a certain frequency, a certain pulse width, and a certain amplitude, and found that, by 4 months, people's seizures were reduced by 40%, and in the placebo group, they were only reduced by 14%. So this is basically and this is, a double-blind randomized controlled trial in which, the patients and the treating physicians don't know what the settings are, whether it's turned on or turned off. So that's the double-blind part. And then they were randomized to be either in the placebo group or the actual treatment group. So that's kind of the gold standard for how we evaluate people. So based on this study, this got FDA approved, and so everybody just uses those settings. Now, how they chose those settings is a whole another story, but those are the settings. It's basically frequency of 145 hertz, a pulse width of 90 microseconds, and then whatever amplitude people want. That's not going to cause side effects. So you could imagine that not exactly personalized therapy, right? It's just everybody kind of gets the same settings, and that doesn't make a whole lot of sense.
And
So
how did they come up with those figures? Sorry to interrupt.
which figures?
The amplitude, the ideal settings.
Based on animal work, basically, and pilot studies from other groups, essentially. Yep, that's pretty much it. And a lot of it is based on, so there's a long history in movement disorders of using high frequency stimulation. Now, obviously, it's a totally different disease process, right? Why would deep brain stimulation for Parkinson's disease translate to epilepsy? That doesn't really make any sense. But they knew those settings were safe, right? And there was a lot of animal work that showed that if you use high frequency stimulation, you can reduce seizures in animals. So it's kind of a combination of knowing that it's safe from prior work in people, and some sense that it worked in animals, and then in pilot studies in epilepsy patients and humans. So
Thank you for explaining that.
There's so do seizures happen in the motor function part of the brain, or can it just be anywhere?
Anywhere.
Really? Okay. So there are parts that you just can't take out, and that's where you bring in this.
Very frequently. Very frequently.
Okay.
There, I mean, so, epilepsy is it's a very heterogeneous disease, right? So one, it can happen anywhere, including in areas where if you took that portion of the brain out, somebody would be unable to move their arm, or, can't see in a half of their visual field or whatever, those are called eloquent areas of the brain, right? So those are parts of the brain you just can't take out. That's actually relatively rare, right? There's a few select areas that you really can't take out. More commonly, they tend to be in the temporal lobes. So the circuit of Papes is it's kind of worked out a long time ago, and starts in the hippocampus, which is the very medial part of the temporal lobe. The outflow pathway goes through the fornix into the mammalian bodies here, which then project to the thalamus, then project to the cingulate cortex, which is in the frontal lobes, and then back out, back down through from the thalamus back to the hippocampus, and forms like a circuit. So this and so the temporal lobe is actually probably the most common area, especially the mesial part of the temporal lobe, the hippocampus, which is one of the more conserved parts of our brain, is probably the most common area, at least in adults, that we see seizures coming from. And so you can resect a large portion of the temporal lobe without giving folks deficits, and that's probably the most common surgery that we do. But the more common thing really is just that either people have seizures that are coming from different parts of their brain, so you can't take out multiple different parts, or they just can't be localized very well with our techniques. The way that we localize is based on either scalp EEG, where we're recording seizures from the scalp, or if people are willing to do it, we'll put in electrodes, actually a bunch of different electrodes into the brain, and then bring them into the epilepsy monitoring unit and wait for them to have seizures, and try to figure out, based on the recordings from the electrodes in the brain, where the seizures are coming from. But obviously there's a sampling issue there, right? You can only put so many electrodes in, you can only record from so many different places. And so sometimes we can't localize it down to a small enough area to be able to take, to be able to resect that portion of the cortex. So the rationale for doing DBS is you can affect probably the most commonly affected circuit, seizure circuit, by putting an electrode right in the middle of that circuit in the thalamus here, in the anterior nucleus of the thalamus. So that's the rationale for neuromodulation, basically. And the idea behind optimization is basically we kind of have this one size fits all mechanism for stimulating people, but there's probably a better way to do this, and we're trying to do this with basically optimization. Optimization is a form of closed loop feedback, in which basically we're trying to take a physiological signal and use that signal to select what stimulation parameters we want to use in DBS. So which signals can we use? Well, we're kind of limited, essentially, right? Because we have 2 electrodes in the brain for deep brain stimulation. So that's what we got. So we can record off of the deep brain stimulation electrodes and use the local field potentials. Local field potentials are basically just a summation of all of the neurons in the surrounding area of those electrodes, to use those signals to try to select stimulation parameters. So the hypothesis behind this is basically we want to find individualized, kind of personalized settings that suppress the neural activity in the thalamus. And the idea behind that is when you look at, if you look at the local field potential signals in the thalamus at baseline, just before a seizure, during a seizure, and after a seizure, it kind of forms this pattern where the overall kind of broadband activity in the thalamus starts to increase before a seizure happens, is very high during the seizure, and then kind of goes back down after the seizure. And so the idea behind this is to push that baseline as far away from, as far down as we can, to reduce the power as far down as we can, to make it less likely that this person's going to have a seizure. So that's the idea behind it. And we have a study kind of set up where it's a 16-month study. Folks get implanted on day 0. They come in for a 1-month visit where it gets turned on. And over the first 4 months, it gets kind of tuned. The amplitude gets tuned by the treating neurologist. Every 2 months, we have to basically download the data. The battery only has a 2-month, can only collect data for 2 months at a time. So that's what these little icons are. And then starting at 4 months, basically, we will essentially, we're kind of doing our own blinded trial where we test out a bunch of settings between 1 and 4 months, pick the settings that look like they're going to suppress the neural activity the most, and then put them on either optimized settings or clinical settings for 4-month periods of time. And so they get essentially 8 months of optimized, what we're calling kind of personalized, individualized settings, and 4 months of the standard clinical settings. And then we're going to compare how patients do, both in terms of seizure frequency, but really just in terms of whether or not our optimized settings can do what we say they're doing, which is suppressing the neural activity in the thalamus. So that's the idea behind this. Those are the outcomes we're looking at, which I just kind of mentioned. It's kind of what some of the data looks like in the clinic. We will test, 9 different settings. The algorithm will then, or the program will kind of then tell us where the hot spots are and where the cold spots are in terms of how much activity there is in the thalamus there. And then we have an algorithm that will kind of sample the available parameter space in terms of frequency and pulse width, and just keep iteratively testing to find wherever the activity, the neural activity in the thalamus is lowest. And then it'll choose a couple settings to try out at home, basically. We don't have to look through this. This basically tells the same thing. There's a lot of challenges with this. Between visits, patients, even just the baseline recordings are not necessarily super consistent. So this is a relatively complicated thing to do. But and just to show you a little bit more data on this, this is what a seizure looks like at home, where we get these at-home recordings where the electrodes are recording at home. We're only getting a 10-minute average of those recordings. We get 1 data point every 10 minutes, but over long periods of time, you can see, these oscillations and neural activity at home. And when you see this huge spike in activity, you can see that's when a person had a seizure at home. So you can actually record seizures on the device at home and correlate that with their seizure diaries, things like that. And when you start analyzing this data, you can start to see this kind of ramping up of activity at home over time, right before they're going to have a seizure. So this figure on the right basically aligns all of the seizures together and then looks at the at-home data. And you can see, in the 1 day prior to them having a seizure, you can start to see this general just kind of increase in the neural activity in the thalamus at home. So the idea would be, if you can see this, to try to ramp up the stimulation beforehand and try to prevent these seizures from happening. And this is just a figure showing that you can really dramatically change the activity. So if you don't have to really know what this is showing, but basically the yellow is basically the magnitude of power. So the scale is yellow to blue. Yellow is much more activity in the thalamus. Blue is much less. And this is the day that we changed their stimulation settings, and you can see it just almost shuts off the thalamus. This particular set of settings really worked quite nicely, where their activity just totally shuts down. And actually, you can see 1 week later, the clinician wanted to kind of go back to the standard clinical settings, and you can see the activities here starts to ramp back up. And the patient actually asked to have it, to turn it back to our optimized settings, and then we turned it back right here. And you can see it decreases again there. So anyway, it's just, we're doing something with this. So in terms of, is this project interesting, or whether or not what you could do if you were interested in it. I think if you're interested in translational research, kind of taking data from the bedside and or from the and using that to inform clinical decision making. If you're interested just in neuroscience or brain signals and how to analyze them, interested in patient care, then getting involved in this might be right for you. I think the goals would be kind of learning the basics of how to collect and analyze the brain signal data, kind of like what I was showing you there, and the basics of how we interact with patients in a research setting, in a clinical care setting, and kind of navigating that process. So that's the epilepsy optimization project. The Parkinson's optimization project, very similar, right? Except there's one key difference, and it's informed by the disease process. So in Parkinson's disease is manifested as changes in movements, right? So it's a neurodegenerative disease process where the dopamine neurons in the substantia nigra die. But we really know that it's actually a lot more than that. It affects their whole body. It's a system-wide process. But when we think about the movement part of Parkinson's disease, it's manifested as a resting tremor. So when folks are just sitting down, they'll notice a resting tremor in their hand or arm or leg, or wherever, most commonly their arm. It's manifested by slow movements, by smaller movements, by stiffness in their body and their muscles. And so when we're thinking about optimizing stimulation for patients with Parkinson's disease, is kind of different, because you can actually measure all of those things pretty easily with things like accelerometers, IMUs essentially. And we have IMUs, we wear them all the time. We have sensors on our wrists. Our phones have accelerometers. Everything that we wear has IMUs in them, basically. And so it's a fundamentally different thing, because a big part of what we care about in Parkinson's disease is optimizing their movements, trying to help people move better, so they can walk faster, so they can take bigger steps, so they don't fall, so they can move more smoothly. And you don't need intracranial signals for that. You can ask people to wear sensors on their body to quantify their movement state. But the idea is the same. How can we figure out DBS parameters to optimize somebody's movement state? Now, the only issue is that in order to optimize their movement state, you gotta kind of know what they're doing. So you have to have some system that can tell you what somebody's doing at home, right? So in other words, if you want to try to optimize, if you want to try to change the DBS settings to change to improve somebody's gait, well, you got to know when they're walking, right? And when they're sitting. So this project is focused on that part. And we're just now moving into the selecting DBS parameters part. But so this project was done by Ali Nuriani, who's a mechanical engineering PhD student that I worked with, that I co-mentored with Raj Rajamani, who's a mechanical engineering professor here at the U. And basically what we did is we sent people home with 5 wearable sensors that they wore kind of just underneath their clothes, with straps, 1 on their chest and 4 on their legs. And we just had them wear them at home for a week. So they charge them at night while they're sleeping, and they wear them all day while they're walking around. And during 1 hour of each day-ish, we have them wear this camera around their neck, and they just wear, they turn on the camera for about an hour. And you get these videos basically of people walking, doing the dishes, moving around their house, stuff like that. And the reason is Ali basically kind of created an algorithm that can tell you what it thinks somebody's doing. Are they walking? Are they sitting? Are they transitioning from sitting to standing, standing to sitting? Have they tripped or stumbled? Turning, a bunch of different activities. And this is kind of just his way of doing this. And but we need some way of validating what, if the algorithm tells us there, and he did this based on just on himself, right? He put these sensors on himself, got him doing a bunch of different things, and said, okay, we can use all the data from the different sensors to figure out what we're doing. But we need some way of validating this. And so that's where the camera comes in, basically. So, 16 hours of the day, the algorithm's telling us what we think the patient's doing. And then for that 1 hour of the day when they turn the camera on, we actually just went through and manually validated what, or annotated the video. So, you can see like during this, whatever, 30 seconds, we annotated the video is the orange and the blue is what the algorithm predicted. So during this 1 minute of data here where it's zoomed in, somebody rated, saw the patient was standing, then they moved to, then they turned around, and then they bent over, right? And each one of these categories tells you what they're doing. And then you can see that the algorithm pretty much overlaps pretty nicely with the manually annotated segments. It's not perfect, right? There's a slight lag here when they move. And there's one section here where it thought they were walking when they were actually standing, but it's pretty close.
And so the cameras were primarily to sort of cross-reference the effectiveness.
Yep, you got it.
Okay.
That's exactly what it was. It's for us to show that, because otherwise, the algorithm just say whatever it's saying. We need to kind of validate it by manually kind of annotating video, essentially. So that's what we did. We got this video software where you can just click a few buttons, and we just kind of customized it so we could, for our own purposes, basically.
I was curious, do they display symptoms in sleep?
Yes, but so Parkinson's patients have or a high percentage of them have what's called like REM sleep behavior disorder, where they will actually kind of like act out their dreams. So there are manifestations in sleep, but not like it's probably just part of the disease process, yeah. I mean, it would be great if we could actually have them wear it during sleep, because I think there's probably a lot of interesting stuff happening during sleep, but maybe not so much manifested through movements. Maybe more stuff that you could see on like EEG or something like that. But yeah, but we need the IMEs to charge, so I think that made the most sense. Then we created these kind of confusion matrices where essentially, we're comparing what actually happened here to what the algorithm predicted, and you can kind of see, it's very good at predicting walking. It's like 99.4% of the time, it's correct in terms of predicting walking and standing. It gets a little less accurate for more complex movements. So turning and bending, it's, the error rate's a little bit higher. Same thing for stand to sit transitions or sit to stand transitions. The main thing are these near falls, where we're really interested in capturing these kind of stumbles or trips, because we think they can predict whether or not somebody's going to fall in the future, the frequency of them. So capturing these is really important. They tend to be rarer events, and so it's not quite as good at those, it predicted like 21 out of 25 of the events correctly. So still pretty good, but and better than anything else out there. There's not really a ton out there. And so, you know, we tested Ali's algorithm versus a bunch of kind of standard machine learning algorithms out there, and it works very well. That's his is the red line. The other ones are kind of a lot of the other standard ones, which all work pretty well for simple things like standing and walking, and a lot of that's already out there. But again, for the more complicated movement events, you know, his algorithm worked really well. So, and I won't go through this, but the idea with this is we want to try to predict future falls, and a lot of the metrics that we can calculate from the sensors. There are a few of them that can predict future falls really well. And then we can create these figures using the data at home versus what we collect in the clinic. So, you know, there is some sense that like when we do gait and balance testing in the clinic, that is not necessarily translatable to home testing. And so, you know, these figures basically look at when we're testing balance in the clinic, we basically pull people backwards and see how big a step they take and how many steps they take. And so you can quantify like how hard you pull them backwards. This is their chest acceleration on the x-axis here, and the y-axis is how big a step they take. You know, and so the slope of that line, like how, you know, how well they're able to match the size of their step in relation to how hard they're being pulled is a pretty good indicator of how well they will respond. And so if you look at people who have a normal pull test, it's called the pull test, they have, you know, kind of like this looking slope, whereas people who have a less normal pull test kind of flatten out here. Anyway, you can look at that versus what they look like at home and how translatable, you know, the clinic testing is to home testing, because we get a bunch of these stumbles at home, and you can see how big a step they take in relation to how far they fall forward or how fast they're moving forward at home. And that's a more kind of ecologically valid way of looking at it, right? Because you know, when you're testing somebody in the clinic, they're kind of ready for it, right? They're ready to be knocked off balance, but at home, when they're just walking around, they're not. So, and actually, it's pretty closely translatable. So anyway, that's the kind of data you can get from some of this stuff. So again, this is translational type of research. These are different signals. It's more movement kinematics, but you know, a lot of the process is the same. There is a lot more machine learning techniques that we use in the algorithm development for this kind of research. So machine learning is something you're interested in. You know, this might be more for you. And again, it's learning the basics of how to collect the data, analyze kinematics, annotating the videos, and then the basics of machine learning techniques are a reasonable thing to be able to kind of get exposed to in a couple of months. I don't know how much time do I have? I have like another. I don't want to go way over time or anything, but
No, you're good.
Okay. So the second branch is health equity research and neuromodulation. I won't go too much into the background since I've already told you kind of what neuromodulation is. But what I will tell you is that our group and a bunch of other groups have done a lot of prior research using these big national data sets that basically show that there's disparities in all of medicine, right? But particularly in neuromodulation as an elective kind of surgical procedure, who gets these is not kind of equitably distributed, and it's slightly different depending on if you look at Parkinson's disease, essential tremor, epilepsy, but there are disparities that exist in all of them. So I'll go through specifically epilepsy and Parkinson's disease, I think is what I put in here, because that's the work that we're doing. So just again, as background, if you look at, this is from a paper we published a few years ago. If you look at the number of overall epilepsy admissions over time, this is from 2006 to 2016, it's kind of like linearly increased. If you look at the number of surgeries produced, or the number of surgeries that have happened over time, there's kind of a corresponding increase until around 2010, 2011, and then it kind of flattened out. A lot of people have seen a similar kind of trend. Basically, number of epilepsy surgeries has plateaued, even though we don't think the epidemiology of epilepsy has changed. The incidence of epilepsy hasn't really changed. And so a lot of people are kind of wondering, like, why are we seeing this? And if you look at the factors associated with who gets epilepsy surgery in the United States, this is only in the United States, by the way. The two major factors are insurance status and race. So if you have private insurance, you're much more likely to get epilepsy surgery. So if you look up here, Medicare patients get epilepsy surgery at about 60% of the rate of people with private insurance. Medicaid patients get about, they're about 25% less likely. Patients who are Black were 45% less likely to get epilepsy surgery than white patients. And those are the two biggest factors. If you look at time, as we saw, slightly less likely to get epilepsy surgery over time, but that actually wasn't significant. One individual period was not significant, but overall it was. So similarly for Parkinson's disease, the overall number of Parkinson's disease admissions has increased over time, particularly over the last 6 to 7 years. And that's probably just due to the aging population. In America, the baby boomer generation is kind of aging out. And as a whole, the demographics of the United States are getting older, essentially. And so you see both an increase in just admissions for Parkinson's disease patients and an increase in the number of patients who get surgery, get deep brain stimulation surgery. Again, I think this is probably just a function of demographics. If you look at who is more likely, or what factors are more or less likely to be present in people who get deep brain stimulation surgery for Parkinson's disease, people who are less likely are people who have more comorbidities, who are older, who are women, and patients who are Black. More likely to have surgery, private insurance, and increasing income quartile. So that's what these are here. This is odds favoring not having DBS and odds favoring DBS. So as your income quartile goes up, you're more likely to have surgery. If you have private insurance, you're more likely to have surgery. You're less likely to have surgery if you have these factors, right? So kind of a similar story as epilepsy, although there are more factors. For epilepsy, income quartile was not really a factor. So the motivation behind this research is basically, okay, these data sets are great for kind of characterizing the problem nationally, but the solution to these problems are going to be more local, right? They're going to be focused on what you can do in your own health system, in your own community. There is a database, an internal database through the Center for Quality Outcomes Delivery and Evaluation, which is called CCODE, which gives us access to kind of our internal data set to see, okay, we know that what the factors are on a national scale. What are the factors within our system that are affecting people who get neuromodulation surgery here, so we can try to devise a plan to kind of more equitably distribute neuromodulation surgery in our own system. And I can tell you, I'll show you the data, but I can tell you that there are a lot of patients. It's almost certainly all referral based, and so where you get your referrals from dictate who you operate on. And as you know, as a neurosurgeon, all our referrals come from neurologists, usually neurologists within our own system, and they get referrals from general neurologists or primary care physicians in the community, and it all kind of gets funneled down. And so who those people are referring are the people that you see and the people that get operated on. And I'll show you a little bit of the geography of that for epilepsy patients at least. So this is just kind of how we use the internal data set. There's like, 3.5 million encounters in it's basically a data set of every inpatient admission in the University of Minnesota Fairview system over the last like 12 years. And that's about a little over 1 million people. And so we use billing codes basically to find people who have a diagnosis of epilepsy who are admitted, not just for surgery, just admitted at all. And there are about 11,000 of those people. And about 130 of them had surgery, and 6,000 of them did not have surgery. And another 4,000 were just people who came in multiple times, basically, which is not surprising. People with epilepsy come in with seizures a lot. We don't have to really go through this, but we kind of went through manually through the charts to make sure that the billing codes we're using to define these people are actually truly epilepsy people, or people who had epilepsy and people who had epilepsy surgery. And so if you do kind of the same thing as we did with the national data set, but we look at our own internal data set here, what we find is people who are more likely to get epilepsy surgery here are younger. They are more likely to be men. This is actually flipped. So they are more likely to be white. So people who identify as Black or any other, we didn't have enough, there weren't enough surgical patients in the other race categories as defined by the US Census to subcategorize. There's just not. But so that's kind of why we have this other category, which is annoying to use, but we're kind of limited by the data set here. They had lower odds ratios, but that actually wasn't significant. And people with a history of depression were more likely to get it. I can go into that separately, but that's a separate category. But the other interesting thing was that people who were in the fourth ADI quartile. So ADI is something that stands for Area Deprivation Index. It's a measure of kind of socioeconomic status. And essentially, the reference value is 1. We split them up into 4 quartiles. And so the lower numbers are people who are in less deprived areas. What is deprived area? Well, it's an index of the income of that neighborhood, the crime rate in that neighborhood, the poverty rate in that neighborhood. They use a bunch of different economic and social status indicators from survey data, mostly from the American Community Survey, to create neighborhood level data to give you a sense of the socioeconomic status. So people who are in the ADI, the 4th quartile, are people who are in the kind of quote unquote most deprived areas. Those people were actually were more likely to get epilepsy surgery, interestingly. So we kind of replicated some of the results from the national data set in terms of things like age and race, but our data internally showed actually that people who came from more deprived areas, kind of lower socioeconomic status, were more likely to get surgery. So we found that kind of interesting. And actually for us, insurance status did not matter. So if people had Medicaid or Medicare, they were just as likely to get epilepsy surgery as people with private insurance in our internal data set. So we investigated the ADI quartile thing a little bit more. We looked at the geography of these patients. It's kind of a busy figure, but I'll take you, it's actually pretty straightforward. So the scale is basically this. People who had a higher ADI, so were coming from more deprived areas, were coded, and this is basically zip codes essentially. were coded as red. Lower ADIs were coded as white. And then the surgical odds ratios, if you were less likely to get surgery, you're coded as white, or more likely to get surgery, you're coded as blue. And so areas where people were more likely to get surgery and also coming from kind of more deprived areas or lower socioeconomic status areas are in purple, right? People who are, you know, not likely to get surgery and lower ADIs were in white, and people who are less likely to get surgery and coming from more deprived areas are red, right? And people who are or more likely to get surgery and coming from more well-off areas are in blue. And so basically what you see is all the blue areas are kind of these like surrounding suburban metro areas, right? So these are like less deprived areas and more likely to get surgery, right? We don't have any purple areas really in the metro, essentially. Maybe I think there's maybe one county here or one zip code. But all the purple areas are coming from kind of more rural regions. And that's basically what we found. The epilepsy patients who were more likely to get surgery that came from kind of more deprived areas were all from smaller towns, basically, in the more outlying, outside the metro, more rural areas, which is kind of interesting to us because by definition, epilepsy patients who are having continued seizures and are surgical candidates can't drive, right? And so they're really traveling long distances to get here. So that's kind of the data that we have here. So this project is focused, and the other thing is all these patients were white. So I think we had, I don't know, there were like 30 or 40 patients who came from these purple districts, and like 29 out of 30 or 39 out of 40 or whatever it was of the patients were white. I think there was, and so the overwhelming number of people who are coming from, you know, more deprived areas or lower socioeconomically status areas who are getting epilepsy surgery were white. So this project is basically a project that is a community-based participatory action research project that is working with this group called Salamo, which stands for Somali, Latino, and Hmong. It's a community group focused on working on doing research. It's composed of community-based researchers from a bunch of different communities within the Twin Cities that look to work with the University of Minnesota to kind of translate research into the community. And because we have such a specific dearth of Black patients with epilepsy, people with epilepsy in our system, both getting epilepsy care and getting epilepsy surgery, that's kind of what the group thought it would be best to focus on. And obviously, the Black community in the Twin Cities is very multicultural. There's obviously a big African American community, but there's also a very big ethnic immigrant community, obviously Somali community, also Ethiopian, Oromo, and so that's what we're focusing on. So this project is looking to basically, we want to create a geographic, a more complete. So I showed you some of the geography, but it's pretty limited. So we really want to create a very detailed geographic data set of people, specifically people with epilepsy in our system and hopefully in other systems as well. So one of our epilepsy neurologists is actually going to Hennepin. So we want to try to include the Hennepin data in that and hopefully create a more complete picture of the Twin Cities, not just make it the University of Minnesota system. And then taking a qualitative approach actually with this Salamo, with this community group Salamo, to look at healthcare, social, cultural, religious concerns that influence that influence Black people with epilepsy, how they obtain subspecialty care, so how they manage their epilepsy, either with their primary care or with a neurologist, and make sure that's kind of culturally specific data that we get, because it's going to obviously differ both within communities and between communities. And then we're working with a health informaticist here, David Haynes, who has actually created this app to look at social determinants of health, and see how that differs between community members. So I mentioned this only because this is really kind of a longer term thing. It's part of the lab, and so I wanted to mention it. But you're not gonna be able to do anything in 2 months, basically, or over the summer. But if it is something you're interested in, you can become part of the group, and just this is like a long-term thing. And so if you're interested in doing this over a longer period of time, then you can get in touch with me and get involved with the group if you want. But not really like appropriate for like a summer project, I don't think. I have like just 2 minutes left, so I'm just gonna tell you very briefly about the last project that you could get involved in. Like I said, I'm interested in gait and balance and movement disorders. We've kind of talked about this already. This is like a super difficult thing to treat surgically. I'm basically, what I'm doing is incorporating a kind of quantitative approach to my clinic visits. So when I see my Parkinson's disease or my hydrocephalus patients in clinic, we basically put this portable motion capture sensor system on them. It's like 15 sensors, a bunch of different straps. You can kind of see what it looks like here. And we run them through gait and balance testing during every clinic visit, so I can get a more quantitative sense of how somebody's doing from visit to visit. And you can get kind of data that will show you how they respond. Remember I said we pull people backwards to test their balance. Well, when you pull somebody backwards, their body, how fast their body moves backwards in space, and it tells you what they look like. And actually, the faster they move and the faster they recover, the better they are. So these are basically people who have a normal response, and then the more abnormal responses as you go down here. And so you can track that over time. And I have a lot of these patients coming through, and there's tons of work to look at this data over time. So if you're interested in again, kind of a common theme here, translational research, looking at movement signals, how to analyze them, how to interact with patients during clinic visits and things like that. And you can get involved in that project too. So obviously there's a huge team, multiple different people, just to acknowledge all the work that they've done on the epilepsy project. I am co-PI with Tay Netoff, who's PhD in biomedical engineering. Zach basically is a grad student, his PhD student, has done almost all the work. Hafsa is a postdoc who has done a lot of that work as well. Yang, James, and Tommy work in my lab to help coordinate everything. That work is funded by the NIH through a UO1 mechanism. The wearable sensor work, as I mentioned, I showed those folks before, is mechanical engineering. And also I collaborate with people at the Minneapolis VA, mechanical engineer and a physical therapist on that. These are the folks from that are involved in the disparities, the health equity work from Solamo and from IHI, David, Kathy, and Shannon, and that's it. So thank you guys for listening.
That was great. Thank you for that presentation.
Sure thing. Obviously, if people have questions too, I'm happy to answer.
Questions from students?
Yeah, I actually have a question. I think the research you're doing is really interesting, especially pertaining to what I'm studying. But I had kind of two questions about your path to where you are now. So why did you decide to pursue neurosurgery? And then also, how did you become interested and involved in the research you are now?
Yeah, I actually did not know I wanted to be a neurosurgeon until maybe like my third or fourth year of medical school. Like there are some people like that I train with that, knew they wanted to be a neurosurgeon when they were like 5 years old. I'm not one of those people. I don't think I mean, you can be, that's great. That's cool. You don't have to be. I always just knew I was interested in the brain. I knew that I was interested in neuroscience and like just kind of how the brain works and what made people do things, the way that he did. And then as I started to learn about disease processes, I got involved in college. There was one of my actually a chemistry professor who did Parkinson's disease research, and it was just super fascinating to me. And I thought that I just wanted to learn more about it. And then in medical school, most neurosurgeons are usually kind of choose between, other surgical fields like orthopedics or, because there's a lot of obviously overlap in surgical fields. But I was just super focused on neuroscience, and I actually was deciding between neurology and neurosurgery. And ultimately, I decided on neurosurgery because I felt like I could do a lot more. I could make, not that neurology can't, but I felt like neurology is a lot of diagnosing and not a ton of like intervening. I mean, there is more and more, but I just felt like as a neurosurgeon, I could always be kind of on the forefront of doing new stuff and making more of an impact in people's daily lives. And I knew that for me, I needed that kind of like daily impact to just, for my own personal satisfaction. It's probably selfish, but I just knew like I needed that to kind of keep me motivated. And I kind of figured that out during my third and fourth years of medical school when I was really getting more involved in patient care. So, and then research, I've just always had this interest in Parkinson's disease since college. Um, and, um, and movement disorders kind of have fascinated me as I've gone through, like how small, changes in one, you know, small system in the brain can have these wide ranging effects, and how that affects people and how you can, just put an electrode in a very tiny part of the brain and have these massive changes in people's movements and their lives. And so I'm interested in just how all of that works and trying to decipher brain signals and figure out how to improve these processes. So that's kind of where I came from, the movement disorder side of things. And um, epilepsy, I I became interested in research because I'm very interested in it from a clinical perspective. They're very satisfying surgeries to do, like the resections. Just anatomically, you see a lot of it's one of the few things now where everything's moving to minimally invasive and smaller, and it's one of the few surgeries where you can really see the brain kind of wide open, and you can, the resections are really kind of like beautiful anatomically, and then you can really just totally change people's lives. And if you can get rid of their seizures, their quality of life is just like so much more improved. And a lot of these people are super young, they're 20, 25 years old. You're altering the course of somebody's life when you can do that. So I've kind of stumbled into epilepsy research because I'm interested in epilepsy as a clinical phenomenon. And then the health equity work is just like, again, if you told me 10 years ago I'd be doing community-based participatory research, I don't know. I don't think I would have known what that was, but it's just kind of evolved from just a sense of like doing, what's right and trying and just being involved in just, kind of making connections with people since I've been here, basically, so.
I think
Thank you.
one,
Sorry.
sorry.
Sorry, I just wanted to say thank you. That's like really insightful.
Sure. Yeah, absolutely.
I was just going to say, I think one thing that was really fascinating about the research is sort of the community aspect that you guys were able to bring into it.
Yeah, I think there's tons of resources here that you won't know about unless you kind of talk to people. And I think there is more of an emphasis, especially in research now, with getting input from patients, getting input from the communities, and not having research be so focused on the researchers, what the researchers think are interesting, right? Because lots of times there's a huge gulf between what patients care about and what researchers think are interesting to be investigated, right? And so there's a big emphasis now at NIH and all the funding places, I think, in terms of kind of bringing that together. Because, and so I think in neurosurgery, you kind of get both aspects. A lot of researchers who are coming from a pure PhD background, they want to know what patients are facing and think is important because they don't have that background. So as a clinician, you can kind of really bridge that gap a little bit. So that's what I try to do. But yeah, and I think the community like specifically focused work on like, that's a little bit different in the sense that like it's really focused on like trying to remedy an ongoing kind of situation. So you're going to see more emphasis on that in the future, I think, in terms of getting communities involved in like the design of the research, the focus of it, all that stuff. Yeah.
Thank you. Any other questions from students?
Yeah, I had one more question. So what would your advice be to anyone who's interested in practicing medicine, but also doing research, like yours that can help out people and bridge gaps when it comes to things like racial and wealth disparity?
I think just like get involved, from an early time point. I think if you're interested in medicine, medical schools really look for I think commitments no matter what it is. They don't really want to see you involved in like 20 different things to like add things to your CV. They want to see, in my opinion, I think, I'm not on a medical school admissions committee, but this is my impression from talking to people and from mentoring students. They want to see a commitment to whatever it is that you're interested in. So if you're interested in medicine, in neuroscience or not neuroscience, or just whatever it is, or disparities, health equity work, then find a group and start working with them, and then maintain that commitment through undergrad and gap years if that's what you end up doing or whatever. And then you get letters from those folks that can attest to all the work that you did while you were there. And hopefully you'll have some kind of finished product at some point, that you can point to, like, I worked on this and we did this and we had an event or whatever it is. But I think committing to, a couple or a few things, depending on how much time you have and how much you're willing to work, and maintaining that commitment, seeing projects through, that's going to be your best bet for kind of getting into medical school, and then just developing your own interests. You might find that, you do something for a year or two and it's not really your thing, but you can still say you did it and then move on to something else. Like I said, like I didn't know that I was going to be doing this kind of stuff 10 years ago, but your interests evolve and your, the meaning that you find in your work evolves too, right? So I think just using the resources you have around you. You guys have a ton of resources here. There's lots of these programs you have available. And the other thing, I think, find a good mentor. And it's easier said than done, right? But find somebody that is interested in mentorship and can kind of help you navigate that as you go through. And those are the two most important things, I would say.
Great. Well, I want to thank Dr. McGovern for his time. hopefully we'll be in touch soon with some research student assignments and yeah, it was really
Cool.
great that you spent some time kind of going over your research and kind of took your time to give our students advice.
Anytime. Always, my email is up there, and I'm sure, Liz has my contact info. So again, even if you're not going to be working the summer or whatever, I'm always happy to help out when I can. So
All right. Thank you so much.
nice to meet you guys. Bye.
Thank you.
Thank you.
Thank you.
I can't imagine opening somebody's brain and looking at it, but it sounds fascinating.
It sure does.
I think one thing that I probably need clarification on is where does the minimally invasive part come in?
I think it depends on the procedure. So, you always want to be minimally invasive as you can, but some procedures don't allow for that. So they might start off with something that's minimally invasive. And if it doesn't work, or if it's not going to be effective for that patient, then they have to go into something else. All right. Liz, let's say you shared a document.
Yes, I have shared a document.
You have hosting power, so you can share your screen at any point if you want. But I'll turn it over to you because I think at this point, it's kind of your show.
My show. Okay, great. So, I shared this document with everybody. It can be found in the student materials folder, but I just want to make sure you're aware of it. This is where all of the in-person location information will live. So there's a couple of sections. You'll see at the top is the research and lab instructor meeting information. So each faculty that's involved in this Pathways program is listed here. And this is their meeting information. So you'll see the room location, the address, any directions and parking information, as well as a contact number if that's applicable. And so I will also make sure to include this information on the calendar invite descriptions that are shared with you as well. So you'll be able to access it in multiple locations. If there's something that changes, you will also be made aware of that. So I've also indicated under each faculty's name what institution they're from, and also if they're research faculty or if they're your instructors. So depending on which lab you ultimately get to be involved in for the next few weeks, this is where you'll access the location information for that lab. A little bit further down on page 2 is site visit information. So on Friday, June 28th is our site visit to the 3M Innovation Center. That's led by Dr. Renu Kumar from Minneapolis College. Here's some schedule information and also directions and parking information. In addition, we will be visiting the St. Anthony Falls Lab on Wednesday, July 3rd. This information in terms of directions, all of that are listed here. The last site visit is for the Hormel Institute. As a reminder, this is optional because it is an all-day visit, but I just wanted to share this with you in terms of record keeping and keeping track of everything. So that is where you can find all of the important details for in-person meetings. Does anybody have any questions about this?
Did you get Amy Larson's?
Yeah, so I have Amy Larson down here.
Maybe I just missed it. Okay.
That's okay, sorry. Yeah, so here's Amy Larson. So I have notes as well. So for Dr. Wenzel, he's working to get parking permits, and I'll provide more details when that becomes available. But we have some time until we're going to Augsburg for that. So, but we'll get it all figured out and available for everybody. If you notice, oh, sorry, did I interrupt somebody?
I just had a question for some of them. It says building access, we need a U card. Is that something that we have to make, or does someone
Yeah.
open the door for this?
That's a great question. Thank you for mentioning that. Yes, so some of these buildings do have access restrictions. So I simply put that here just to make you aware that you might have some difficulties getting access in the buildings. However, the research faculty know this, and we're going to coordinate so that someone will be able to let you in the building. So some buildings have public access and some do not. So just an FYI, but we'll make sure that you get access to, that you are able to get into the building, but we'll not be able to give you a U card. Does that answer your question?
Yes, thank you.
Any other questions about in-person logistics?
I'm just reading through the document. It's very comprehensive. So thank you. Liz.
Yeah, I know it is very detailed. Feel free to email me if you have any other questions. I'm happy to help clarify anything. I tried to give you as much detail as possible that you would need to go to these in-person meetings. Okay. And as a reminder, looking at our tentative schedule, you know, it sort of goes hand in hand with both of these documents. So you'll be able to keep track of where you're supposed to be and on each day. Okay. Any questions before I move on?
No, but I do want to note, so if we've been using my Zoom address for consistently throughout. Some of
Yeah.
these meetings with other faculty might have a different Zoom address. We will try to ask them to record it and provide the file for us. Or at least if something else, they can access it, but if they can provide me the file, I can put it up on the YouTube channel. Because usually typically it's just an MP4 file, but you may have a different Zoom link. Double check with Liz's invitation because she's really great at making sure that the Zoom link is in the invitation. So if you're not sure which Zoom link to go to, it should be right in your invitation on your calendar.
Yes, thank you for mentioning that too, Jessica. So, for example, on next Friday is a virtual session, but Dr. Joan Pfeiffer is going to be your instructor that day. So she might have a different Zoom link, but we'll make sure to update the calendar invite and any other materials. And I'll also send an email the day before, just reminding everybody. So you'll definitely have access to that as well. Any other questions on scheduling? Okay, so I'm going to stop sharing, and then what I'm going to do now is I will share in the chat the Google form so that you can make your selections. Give me 1 second. Okay, so there's the Google form. Let me know if you're having issues with it. You should be able to fill it out. Just take a few minutes And then if you're free to go after you're done. But if you could stay on here and complete it, that would be really nice so that we know.
I have a, here, I can stop recording.